Hello everyone. In this module, we'll be talking about the mechanisms of cell injury. Antimicrobial agents can injure or damage microbes by several mechanisms. A detailed knowledge of how an agent kills or inhibits the microorganisms is essential so that the effectiveness of these agents can be increased by suitable changes in the conditions. Study of a typical cell tells us about its important constituents. A typical cell is protected by a well-defined cell wall, followed by a cell membrane, which by its semi-permeable nature controls the inflow and outflow of nutrients. The cell contains a number of important proteins and enzymes which catalyze metabolic reactions. The cell has a number of organelles, each having its respective function. Damage to any of these cell constituents can lead to the death of the cell. The first mechanism of cell injury is damage to the cell wall. Cell wall is the outermost protective covering of the cell. It normally protects the cell from damage due to changes in the external environment. The cell wall of any microorganism can be damaged by the addition of antibiotics. Many antibiotics like penicillins, cephalosporins, and carbapenems inhibit the formation of cell wall, leading to formation of protoplasts which do not have osmotic protection and result in the lysis of the cell. Penicillins mainly retard the formation of peptidoglycan, the major component of the cell wall in gram-positive organisms. Penicillins block the transpeptidase activity during peptidoglycan synthesis. As a result, the peptidoglycan is not formed properly, leading to the cell death. The cell wall can be damaged by the addition of surfactants as well. Substances which lower the surface tension of the medium are called surfactants. Soaps, detergents, bile salts are some of the examples of surfactants. These damage the cell wall by dissolving the lipids present in the cell wall. Cell wall can also be damaged by the addition of certain enzymes. Certain hydrolytic enzymes have the ability to hydrolyze the cell wall and hence bring about the death of the microorganism. The enzyme lysozyme hydrolyzes beta-1,4 glycosidic bonds of the cell wall, and many gram-positive organisms are affected by it. The next mechanism of cell injury is the damage to the cell membrane. All cells are bound by a cell membrane which is selectively permeable. This is the cell membrane. Surfactants can damage the cell membrane by dissolving the lipids present in it. This destroys the selective permeability and results in the inhibition of the growth of the cell. Certain antibiotics like polymyxins act as detergents and distort the bacterial cell membranes. This is probably by binding to the phospholipids present in the cell membrane. These antibiotics are particularly effective against gram-negative bacteria which have an outer membrane rich in phospholipids. Polyene antibiotics such as amphotericins bind to particular sterols in the membranes of the fungal cells and damage them. The third mechanism of cell injury is denaturation of proteins. Proteins are active when they are in their native configuration. Any substance which changes the conformation of the protein results in denaturation or coagulation of the protein and prevents them from carrying out their normal function. This may damage or kill the cell. In denaturation, hydrogen and disulfide bonds are disrupted and the functional shape of the protein is destroyed. If the proteins are treated with mild heat or dilute acid or alkali, they undergo temporary denaturation. When the denaturing agent is removed, some proteins can regain their original conformation. Many antimicrobial agents, however, bring about complete and permanent denaturation, leading to the death of the microbes. If denaturation is temporary, it is bacteriostatic. Denaturation of the proteins can be brought about by hydrolysis. Acids such as boric acid and strong alkalis destroy proteins by hydrolyzing them. Denaturation of the proteins can also be brought about by oxidation. Oxidizing agents such as hydrogen peroxide or potassium permanganate oxidize the disulfide linkages or sulfhydryl groups. Denaturation of the proteins can also be brought about by attachment of chemical groups. Heavy metals such as gold or silver attach to the sulfhydryl group. All these reactions can kill the microorganisms. The next mechanism of cell injury is inhibition of protein synthesis. Protein synthesis involves the activity of DNA, RNA, and also ribosomes. The bacterial ribosome, which is 70S, is different from eukaryotic ribosomes, which is 80S. This difference allows certain antimicrobial agents to selectively attack the bacterial cells. Certain antibiotics, like streptomycin, interfere with the protein synthesis of the bacterial cell. It acts on the 30S subunit of the bacterial ribosome and interferes with the correct reading of the mRNA. This leads to the incorporation of the wrong amino acid. Antibiotics like chloramphenicol and erythromycin act on the 50S subunit and inhibit the formation of the growing polypeptide. The next mechanism of cell injury is inhibition of replication and transcription. The process by which DNA duplicates to form daughter DNA is called replication. Transfer of genetic information from DNA to mRNA is called transcription. In replication, the main enzyme is DNA polymerase, and in transcription, the main enzyme is RNA polymerase. Damage to any of these enzymes would result in the inhibition of replication and transcription respectively. Antibiotics like quinolones inhibit bacterial DNA synthesis by blocking the enzyme DNA gyrase, which is responsible for reducing the strain caused by unbinding of the DNA double helix, which is necessary for replication. Antibiotics like rifampin blocks transcription and kills the organism. The next mechanism of cell injury is inhibition of metabolic reactions. Metabolic activity of a cell is due to various enzyme catalyzed reactions taking place in the cell. Out of these metabolic pathways, which provide energy to the cell if affected, is particularly harmful to the cell. Glycolysis, TCA cycle, oxidation, and oxidative phosphorylation are some of the pathways which provide energy to the cell. The enzymes which catalyze the reactions of these pathways can be inhibited by a number of antimicrobial agents. For example, fluoride inhibits glycolysis. Arsenic compounds inhibits the TCA cycle, and cyanide blocks oxidation. Dinitrophenol inhibits oxidative phosphorylations. So with this, we come to an end of the different mechanisms of cell injury which can be brought about by various chemical agents which are responsible for killing or inhibiting the microbial growth. If you like the content of this channel, like, share and subscribe the channel. I would also like to hear if you are interested in any particular topic for content creation. Thank you.